Hello, my name is Meena Subramanyam, and I will be presenting the module on the interpretation and reporting of immunogenicity data from clinical studies of therapeutic proteins, which are also referred to as biologic drugs, biopharmaceuticals, or biological medicinal products as part of the Immunogenicity 101 series. The objectives of this module is to first provide a framework of harmonized terminology for the assessment and reporting of the clinical immunogenicity of therapeutic proteins and peptides. The module will also describe potential methods for analyzing, reporting, and representing the descriptive characteristics of the anti-drug antibody response and its impact on clinical parameters. The consequences of product immunogenicity vary from no evidence of clinical effect to severe life-threatening responses. Therefore, in order to appropriately assess benefit-risk of the therapeutic protein or peptide and to support safe use of the product in clinical practice, the elucidation of the clinical consequences of immunogenicity or anti-drug antibody responses is critical. A variety of stakeholders are interested in understanding and contextualizing immunogenicity data that is generated during therapeutic drug development. First and foremost, regulatory agencies use immunogenicity data in the analysis of the benefit-risk profile of the therapeutic. In addition, based on the demonstrated immunogenic potential of the therapeutic, the regulators may consider the need for additional longer-term immunogenicity data collection as part of the ongoing safety studies or the risk management plan and any associated risk mitigation measures that may be required to guide patient management. Second, the healthcare providers, physicians, and nurse practitioners who may need to be aware of clinical immunogenicity results. Biologic drug package inserts or prescribing information documents describe clinical immunogenicity to varying degrees. They typically mention the overall incidence of anti-drug antibodies and/or neutralizing antibodies in specific clinical studies, and in some instances also cite the clinical impact or lack thereof of developing anti-drug antibodies. The information generally does not inform physicians and patients about how to manage immunogenicity in routine clinical practice. To provide optimal treatment plans for their patients, medical practitioners may also benefit from having specific information on immunogenicity relating to the safety of initiating a treatment regimen, the maintenance of treatment efficacy with an acceptable safety profile, and clinical management options when ADA develop in their patients. This may enable healthcare providers to utilize immunogenicity data to develop patient management guidelines as well as to determine impact of antibody formation on follow-on switch treatments. Third, the payers. They may also use immunogenicity data to make relative comparisons of products belonging to the same therapeutic class, such as interferon beta therapies or anti-TNF therapies, to understand cost-benefit considerations depending on the clinical impact of anti-drug antibodies. And finally, the marketing teams within pharmaceutical companies may use immunogenicity data for product positioning relative to competitor product.